During the broadcasting, I was so many people, even like walking in the street, the people know me and shout out. But right after that, the broadcasting ended, nobody recognized it. In a headline, department store, so people just我想原先的用意该是里面签的，对，我有没有 point？ 交的报告是不是 OK？ 应该可以。Okay, ladies and gentlemen, we will start within a minute. So after Dr. Silver gives the talk, then we have a lunch here. I know several colleagues have the class right after 1 o'clock. For those who can have time, you can join us for lunch box. And today we have Dr. Silver give us one of very important talk about the first steps of the origin of life. Let's welcome Dr. Silver, Professor Silver.
Thank you, Lee and I. It is 1 month too late to welcome us. We have been here already for a month. But
welcome you.
Thank you. And it feels like home because this is the 4th year here, I think 5th time here. And we are happy, so we thank you. I didn't know what to say today, and so I'll ask some questions. One, it will be a short talk. Yesterday, I talked about an unrelated topic that I've talked in this department last year and spent 90 minutes. Today, maybe 35, shorter. Okay. Have I talked about the first cell here before?
Never heard before.
You've never heard before. Okay. So I have been very lucky. Bert's not here. I say Scott, but I don't see Bert's not here.
No.
Okay, fine. When I was an assistant professor in a department of life sciences like this, nobody cared what I did. So they left me in peace and I could do research. And mostly I taught things that nobody else thought about. And so I've been very lucky in that in addition to our own research, which is microbial, bacterial molecular biology, which I'm going to talk about next week somewhere else as we're working. We've done other subjects that nobody cares about. So I gave the first lecture on HIV/AIDS at my medical school to the medical students 25 years ago almost, because nobody was interested in HIV. The virologists were all too interested. You are interested in your virus. People are interested in their specific, and nobody was interested in HIV. There were no retrovirologists. And so I got to learn about that a little. This is a little the same. I had a friend, his name is Arthur Koch, and he is very good at thinking and terrible at writing. And after more than 20 years, every 5 years of rewriting a paper of Arthur's, he was born in New York, raised entirely in the United States. Americans are often very terrible at writing. He said he was thinking about this subject, the origin of life, for a long time and writing about it. And the manuscripts never could get published. Would I write with him? And it was somewhat different. So 4 or 5 years ago, Arthur and I published a paper called The First Cell, The Origin of Life. And I learned about from him this subject which I never thought about, and I did not do research in my lab. There is research being done now on the origin of life. By the origin of life, we mean how do we go from no cell to the first cell on the planet Earth? Okay. There are two alternatives. I'll say it now, and one put aside quickly, is that life came from another planet, maybe Mars, maybe from another solar system, and came in a meteorite. And this subject is sometimes called astrobiology. Because, you know, life in space. Francis Crick thought about this a lot and wrote about this. He, for a while, thought maybe life came from somewhere else. And there's a technical word for it which is not interesting. But it either came from somewhere else or it started here. I think it probably is easier to think it started here, but there's no data to tell you either way. So I'll talk only as if it started here. Astrobiology is supported by the US government, which has, you know, an astronomy program. They look for life on the moon. They look for ATP on Mars, found nothing. Martian meteor. Oh, please stop with questions anytime you want, because it's very relaxed. Martian meteorites land on the Earth all the time. I don't know how often. And they often have organic materials in them. So people think organic compounds, maybe they came from Mars. Martian meteorites often have small things you can see in the electron microscope that look a little like nanotubes or small bacterial cells. So every 2 or 3 years, there's a report in the newspapers that maybe life has been found in a meteorite. They are always wrong so far, and I think it has happened enough. Probably they will always be wrong. The first report says yes, then all of the people who understand the chemistry or the microbiology say no. This is of interest to the US government because space program, to the Chinese government because space program, and to the European governments because space program. Most of us are completely convinced that there is life somewhere else out there, but only because we think, what should be special here? Since life started here, why shouldn't it start somewhere else? And that eventually we will find it, but maybe not for another 50 or 100 years, I don't know, but probably not this year. If it would be easy to find it this year, we would have found it last year. Okay? So I'm going to talk about what is the first cell, the origin of life, as if life originated on our planet, on Earth, okay? And what was necessary for the first cell. And about half of the PowerPoints are from this paper that Copeland I wrote about 4 or 5 years ago. Again, for background, people who think about this, do you know the name Leslie Orville when you read about the double helix and Watson and Crick? He was the theoretical organic chemist who knew some organic chemistry. Crick knew nothing. Watson knew even less. Orville understood base pairing and tautomerization. So he then spent the next 40 years at the Salk Institute where he worked, studying, doing the origin of life. So there's There's a journal called The Origin of Life. You can Google it, it's the easiest way to find it. There's another journal called Astrobiology. So this is a subject that I have never been at a university that has. So you said, John, it's important. I'm not sure, because no university I've ever been at really studies it. Okay? I have it as a hobby, and because my university in Chicago doesn't care what I do, it's okay, I can do it. But if I wanted to have projects for my lab, I think I would not recommend it. Okay. First, I have a bunch of slides. Again, we'll say, have I shown you this one before? Has anyone seen this one before? Okay, can you see it at a distance? Do you know? So I have a bunch of slides that are tourism for. This is how China, the People's Republic of China, sees the world from the British weekly magazine called The Economist. For Scott and myself, John Few, it's a little bit like the New Yorker cartoons, but it's more on economics. And so this is their cover. And you see here is the Imperial Palace, the Forbidden City. Here it says Tiananmen Square. And then immediately you have the ocean, and here's Taiwan. It has a flag on it. Nothing in the middle. Then there's Japan and the United States. And the United States is at the edge of the water, and immediately the Statue of Liberty and Wall Street, because that is, of course, where China would be interested in. And then I think Europe, okay, and Africa. So this is not quite a joke. The New Yorker ran a similar one on New York City, how New York City sees the world. And New York City, I think, was at the top. And then there was nothing until Los Angeles, and then there was nothing after Los Angeles, because that's how New York City sees the world. Okay. We have been very lucky, Lee and myself, that we have come to Taiwan many times now, about 5 or 6 times now. And we've traveled elsewhere in the world. So I have this slide and another one upside down that I took out today that shows where we visit. And she moves the arrow from here to here to here to here, wherever we are for different talks. And it's good fun. Okay. This is Chicago. I think I would have put this in an earlier, a different talk, but I like to say it again. We live here. The university paid for a helicopter to take this photograph because they were building technology park for companies, for biotechnology companies, and there were 4 or 5 in a building that was built here that were interesting to me, that I had worked with. The last one moved out earlier this year. Okay. Our lab is in this building, so if I go here, Michael Jordan plays basketball here, okay? It's only about 1 kilometer, but there is a highway in the middle, so it's not convenient to walk, okay? So this is the Sears Tower, which is, after 9/11, again, the tallest building in North America. And it is about maybe 6 kilometers from our house. I have walked once from the Art Institute, which is here, Symphony Hall, the Opera House, museums, museums, museums. Oh, did you read, Lee, that they're sending a beluga whale away for mating? We have whales in Chicago. They are Arctic whales from far north that were brought maybe 10 years ago, and they live in an aquarium building. And so they are sending a male to another place where there are whales for mating in today's newspaper. Okay? So here is basic sciences at the university. This is the hospital where we can walk. It's 200, 300 meters. We don't need a bus. But the University of Illinois in Chicago is very, very close to the center of the city, which is convenient and nice. Shanghai University is very, very far from the center of the city. Okay. One of our immunologists likes to sail. Here is the Sears building, the tallest building in North America, from 10 kilometers out at sea on Lake Michigan. This wonderful lake is 200 kilometers from west to east and about 500 kilometers from south to north. So it is very big. That's bigger than from here to China. But it is fresh water, so it doesn't smell right. It doesn't smell like the sea. And there are no crabs or shrimp, only fish. And bad pollution, so we do not eat, I try not, people fish, but I don't want to eat anything from Lake Michigan. But sailing, this immunologist is a very strong sailor, and I went a couple of times. This is not Chicago. We're lucky we traveled. This is a polluted beach from an oil spill in Karachi in Pakistan. I often we talk politics. I think it's important for Taiwan people to go to Pakistan to help them. Pakistan needs help. It needs help all the time. And there are good people even where there are bad governments. I don't like the Pakistani government, but they're good scientists. And I have a friend, she is chair of the Department of Molecular Biology at Karachi University. And we have been north in Lahore and have been to Islamabad. I have about 4 times been places where the next week or the next year there is a bomb and 50 people are killed, but never while I am there. Okay, or I would not be here. This is not Pakistan. We are so lucky. This is Lee about 9-10 years ago riding an ostrich. Do you know the South Africans want to do the ostrich genome? Because it's a good economy animal, because the skin can be made into shoes and belts and women's purses. And I've been thinking the last month which genomes are going to be done in Taiwan, and what is the best genome for the Shanghai genome. China did first rice and then panda when they were very expensive. Now that a genome can be done for really a few thousand US dollars, you can think about what genome should be done at Shanghai University. This is the Department of Biomedical Sciences, and South Africa wants to do that one. I always say, Lee wrote this, it runs around very fast, about 30K. She did not fall off, so she was very happy. They have small men who run around, teach you how. You hold, this is a wing. The wing is just a little bit, it's not a big wing. And of course it cannot fly. Look at how tall it is. And I am not allowed to ride this. You must be 50 kilo or less. So I will never be allowed to ride an ostrich. Okay, but Lee was lucky and had a nice time. I am allowed to ride on this. Okay. And this is again in a unhappy country a little bit further north. We went for a holiday, less than a week, to Zimbabwe, where the terrible man Mugabe is still there. I always say freely what I think, and sometimes I got in trouble yesterday, maybe, by saying something against People's Republic of China, and I didn't know there was a Chinese student sitting there. But I have been doing that since I was a child. I say what I think. So we were in Africa, very lucky. And then we'll get to the first cell, okay? This is an active subject, the first cell, the origin of life. It's being done. Here are 2 PNAS papers that were published this year, okay? And one, the primary author is Sostak. I don't know how to pronounce it, I've never met him. Do you know this man?
Yeah, Shostak.
Shostak. Have you met him? Because,
Yes.
okay.
He was Andrew Murray's PhD advisor.
Oh, I didn't know that.
Yeah.
Shostak. He's a professor of molecular biology at Massachusetts General Hospital, which is a hospital associated with Harvard Medical School. Correct me if I make a mistake. Good hospitals, there's a good tradition. I spent 2 years at Hammersmith Hospital, London, of good science, biomedical science being done in hospitals. And I think that's a tradition we should encourage because it can be good. So Shostak has been maybe 40 years at Mass General Hospital. I think Howie Goodman hired him when he started the Department of Molecular Biology there. And he is, the name is Polish, but he was born in London and raised in Canada. And 3 of them shared the Nobel Prize 2 years ago for telomerase, how we maintain the ends of chromosomes. But immediately after doing work on telomerase, Shostak decided he was going to do the origin of life. So some very interesting people have found this a good subject when they have been so strong that they don't have to worry about grants. I don't think he worries about grants. You never get NIH grants for this. But he publishes in PNAS, and Blackburn, who was the name I associated with this Nobel Prize, she is from New Zealand, but living in the US. Shostak is English Canada, but living in the US. I think there was one person they allowed who was American born. But the importance of overseas people working in the United States for American science, it has been very, very much. And I think the United States has been very lucky. So this one is on nucleic acids. And this one is on membranes, okay? And I will show you that we think we need both membranes and base paired nucleic acids in the first cell, okay? And he has contributed, does experimental work supported by the Science Foundation for both. And I'll go into this in some detail. And these 2 papers were very interesting to me. I read them for today. I had not read them before. And he says, nucleic acids before ribozymes. I'll talk to you about ribozymes. But what would a nucleic acid have looked like before ribozymes? Ribozymes have A and U and G and C and phosphodiester bond 3', 5', 3', 5', like DNA and RNA that we know now. But he says in that paper, if you wish, before ribozymes, maybe there were other bases, and especially maybe it was not 3', 5', maybe sometimes 3', 5', 2', or 4', not always rings, somewhat linear, sometimes ribose, sometimes another sugar. And then slowly we evolved in the first cell, or before the first cell, maybe in the first cell, that ribose is best for a ribozyme. Okay? So that happened afterwards. The second paper, which I read here in Shanghai for today, says maybe before phospholipids, no phosphate. I will show you the note ATP, so it's not hard to imagine no phosphate, just natural hydrocarbons. The meteorites have hydrocarbons, carbon, 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 hydrogen. And then they will make a leaky membrane, he says here. So I think if we do this again next year, this is going to be a new figure, okay? We do it different places in the world, sometimes once a year, twice a year. And then leaky membranes, as they become fatty acids and phospholipids, become more rigid, not leaky. Therefore, we need metabolites like amino acids, sugars, and things inside the cell. Whereas when they were leaky, maybe they just came in and out. And as they are inside the cell, we also need bioenergetics, energy coupling to move across the membrane. But when I wrote the first cell paper 4 or 5 years ago, I had not really thought about this carefully. Here is a paper from this new article, and it said, you know, leaky membrane may be in the first cell, and things can just go across. But then, as we make more familiar proteins, fatty acids phosphorylated at the surface, things cannot go across, so we must make things inside. And sometime in the origin of the first cell, we start making things inside instead of taking things from outside, from the media. If we are E. coli or humans, we still take food from outside. When I was just, maybe, have I shown this one before? Here?
I saw this
You've seen
picture
it.
before, but not
Okay.
from yours.
Not from mine.
Yeah.
This is my version. The idea, or I learned it as a student from Jacques Monod, who did the Lac Operon in Paris, at the Institut Pasteur in Paris. And he drew a picture, maybe that you're referring to, on a napkin at a restaurant. And he said, if you want to understand the African elephant, which is 2 meters long or 3 meters long, then it is best to study E. coli. And when I heard this when I was 22, 23 years old, I said, okay, for the next 100, that's what I'm going to do. And we still study E. coli. I still like E. coli. Okay. The big difference between E. coli and the elephant is on a scale. This is 1,000th of a millimeter, and this is a meter. Okay? They both have sensing and motility and eat, and have DNA, RNA, and protein synthesis, of course. And the DNA, RNA, and protein synthesis are similar. This from, if I've shown sometimes your students, but I'm not sure the faculty here, is how bacterial people like to say against animal people, most of you are animal people, that most of the world is prokaryotic, microbial. Most of the biomass now is not trees, it's microbes. Some, the insect behavior person, E.O. Wilson, Stephen Gould, who was the paleontologists who like to write a lot, they at some stage decided this would be interesting. And animals are only a small thing here. Closest to fungi, even what we used to call protozoans, but don't now, amoeba and ciliates and flagellates, they are more diverse. Animals are all very, very similar. And C. elegans and elephants are in the same small group that are very, very similar. Compared to the diversity at the molecular biology level, 16S RNA, but gyrate, any sequence homologies, where we have many kinds of bacteria and many kinds of archaea. That's not today's project. Today's problem is what started here at the beginning of life, from no cell to the first cell, before there were bacteria, before there were archaea, and of course eukaryotes came about 1 billion years later, after the first cell. I don't know if you've seen this one, which I like to think about. The last one was from a textbook. This is from a publication. And trying to put all life together. And here you have plants, and maybe are called protozoans, I don't know, metazoan. So the animals are very small, and most are prokaryote, which are more deeply related. Okay, so that's a technical. Now we get into the first cell. Okay, the first cell, the origin of life. Before the first cell, there was chemistry going on. Lightning flashed, and maybe some of you knew, I'll show in a minute, the Miller-Urey paper, 1953. And there were amino acids, maybe purines, pyrimidines, small organic molecules caused by non-biological processes, heat, electricity, energy input into the growing planet. And it's here. This was the PhD thesis of a man by the name of Miller, who I think recently died. He was a professor at UC San Diego for most of his life. And in the physicist's lab, Urey, on the origin of life. And then these people thought it would be fun. Stanley Miller's warm soup, prebiotic soup, okay? It goes back even to Darwin. Darwin, who of course wrote about the diversity of life, that we call her, thought about how did life start then? And he thought maybe at the side of the sea, there was a warm soup by processes he never imagined. And now we think they're cosmic processes with high energy. Now some people think it happened at the bottom of the sea where things were very hot and is coming out volcanic-like. So 200-degree water plus high pressure at the bottom, and it cools quickly. There are strange microbes, bacteria, archaea, that are growing maybe 5 kilometers down, and they have to make and in fact there are animals, there are worms that grow on the bacteria, and some of these worms that are 5 kilometers down have no stomachs because they don't need it. They are just filled with bacteria that feed them, and they can be 2-meter long worms. I have never seen them. They only live 5 kilometers under the sea. Okay, so this was origin of life. I still haven't come to the first part of our work. It's an active subject, and I'm going to go through a few slides now quickly because I don't like them. So I'm not going to go through them in detail, but just to say there are different opinions. Not everyone agrees. Koch and I agree, and I think pronounce it again?
Shostak.
Shostak. Similar ideas. I like his ideas. But some people, including Morowitz, who was an older person than me, who's been thinking about this for more than 50 years, he likes enzymes, so he thought metabolism first. Some people like energy. So there's someone called Wachtenschauer, German name. He thinks energy first. But most of us who are molecular cellular biologists, we think nucleic acids first, and RNA and ribozymes. But this, I don't think, is a particularly good picture, and the next 3 or 4 pictures also are not. The people who like metabolism first start having a citric acid cycle before there is life. I think it's wrong, but you would have to sit down quietly and think about it. This is what Pope taught me and I like. At the beginning, there was prebiotic synthesis before the first cell, and it was from electricity, lightning, heat, chemical energy. And we made all sorts of small organic compounds. Like the Earth made small organic compounds. We were not there. Amino acids, sugars, a wide variety, non-specifically because it was done by organic chemistry, no biology. Then we have to get 3 things together: ribozymes and membrane vesicles and a kind of energy to put them together. And these new references might be, tell me here. So if I had to do it again, I think I would add more to that. And then we had the first cell about 3.8 billion years ago, very soon after the Earth started. Life that was a surprise to me. Life has been on the Earth for 85–90% of the time there has been a planet Earth. When we think about humans, we think humans have only been on the Earth a couple 100,000 years, the smallest time. But life has been the longest time. But after life started, things happened very, very quickly, and we got this diversity of microbes in maybe 200 million years or so, very quickly. And oxygen was put out into the air by a kind of bacteria called cyanobacteria. And then we had air, oxygen in the air. And so we could have eukaryotes in animals, and that happened much shorter time. But we're going to talk about the first cell when Darwinian selection can start. What do we need? We need a membrane, we need energy, and we need a nucleic acid. That's all. We do not have any of these things. I'll repeat that again in a minute. After a few 100 million cells, from the first cell, a few 100 million years, we had a cell that is called the last universal ancestor. Research over the last 25–30 years, molecular cell biology, says that we are all the same. We're so similar, remarkably similar. We all have ribosomes, whether we are bacteria or humans, the elephant or E. coli. We all have nucleic acid polymerases, DNA and RNA. We are basically the same 20 amino acids. We're remarkably the same. So they must have all been there by the last universal ancestor. And then there was some kind of a split, which we don't understand at all. And bacteria and archaea are remarkably different. And that's not today's, because today is here. And so they have different membranes, different polymerases. And so, in fact, a bacteria and an archaea fused to make the first eukaryotic cell less than 1 billion years ago. And the archaea was more the nucleus, so that we think, because the nucleic acid polymerases of archaea and humans are more similar, and maybe the bacteria made the cytoplasm, because the bioenergetics and the sugar and amino acid biosynthesis and things like that are more bacterial. And one person, also near Boston, Lynn Margolis, she thinks there were 5 fusion events. Endocytosis, where 2 or 3 or up to 5 different kinds of prokaryotic cells went together to make our current eukaryotic cells. Well, we know because we've been taught it, we don't have to think very hard, that mitochondria are really bacteria that got into us for bioenergetics. Now we know which kind of bacteria. They're proteobacteria, because from DNA sequence analysis we can tell which kind of bacteria. And chloroplasts are just cyanobacteria that got in to start plants. Okay? I wrote, I do not believe this, because there are lots of different hypotheses, and this is a paper about 5 years ago that involves viruses. There are people who say maybe viruses began before the first cell, and that viruses evolved to become cells. Other people, such as myself, we can't prove this. This happened 4 billion years ago. So you do experiments and have ideas, but they can't. It's not like our normal science where we can almost prove things. Here you can agree or disagree. That viruses maybe came very early, before DNA, and that it went from, and that independently 3 times maybe viruses that had DNA infected cells that had RNA to make the transfer from RNA-based heredity to DNA-based heredity, which happened after the first cell, but before the last common ancestor. So I don't believe this, okay? But it's important to know there is a diversity of opinions and people. This is so complicated that who can follow it? And it says a model for the origin of life, but I think it's basically not very useful. This is another one, and I think it's not very useful. And this is useful, but I haven't really thought about it. When did iron come in? We have so many enzymes, so much of our biology is dependent on iron. Iron in hydrogenases. We have iron for sulfur for cages for electron transport. Mitochondria, chloroplasts, E. coli, the elephant, both need iron for sulfur 4 and iron 2 sulfur 2. I'm sorry, this is a poor figure that we captured off internet. But when did iron come in? Iron came in quite early. Copper came later, only after there was oxygen in the atmosphere. That's what people mostly think. Copper was just about the last element to be used by life. Maybe selenium, selenomathione, came still later. At the beginning was, of course, hydrogen, carbon, oxygen, nitrogen. Phosphate came later, the beginning of the first cell. Phosphate, maybe just at the
sulfur. Sulfur must be similar to the species of the ferric iron.
Right. Yes. I think sulfur and iron about the same time. Don't you think?
potential.
Yeah.
Because very
Yeah.
fusion potential, right?
I agree.
Sulfide.
Ferrous, sulfur was involved in early pre-cellular and first cell bioenergetics, I'll show in a minute. Okay, the ideas of the origin of life are not new. Here, 140 years ago, were trees, but the trees that were our grandparents learned before us always started with small, simple cells like yeast. They didn't even think about bacteria. And then come to humans, Menschen in Deutsch, in German at the time. So the ideas that we're talking about were very usefully and well started, I think, say 100, time of Darwin. All of this starts at the time of Darwin. Here again, I still haven't gotten back to much details, and I'm taking more time than I thought I would, was the tree I showed you, which most people, bacterial people, like. They're bacteria, archaea, and eukaryotes, including elephants and yeast. And that it has mostly been diverse, spreading and becoming more diverse. Some people emphasize lateral gene transfer, and here's a cartoon showing it. And it certainly has happened. But I think more 90% this and 10% this, okay? And again, there's not agreement on that. Okay, here's the first cell from the paper that Lee drew the figures a long time ago. We needed three things: a ribosome, energy, and you see the energy we think is sulfur and iron. But no ATP in the first cell. Very strange to think about this. The first cell, everyone agrees, did not have ATP. The first cell, everybody agrees, did not have DNA. The first cell, everybody agrees, did not have proteins because it had only ribosomes, and we can't make proteins until we have ribosomes. Ribosomes are basically large ribozymes. The catalytic part of ribosomes is the RNA. The protein only is structural. So the evolution from ribozymes, as you're familiar with them, to a ribosome comes after the first cell, but came very quickly. And I think no metabolism, no enzymes, except ribozymes. No protein enzymes, no proteins. So this seems to be relatively agreed. The energy of inorganic iron-sulfur energy can separate, so we have protons on the outside more than on the inside. So we have a membrane potential, and we can move electrons across with inorganic process in the membranes. No enzymes. So here we have a bit of a membrane potential, chemiosmosis, like modern mitochondria, but no proteins. Now we use proteins. And I'll show you, we think that little peptides started, and first were not enzymes, but affected membrane permeability. And they started from the end of ribozymes, as ribozymes do now, and of course tRNAs have an amino acid at the end, okay? Early membranes. I don't know how many of you know, maybe all, that not all cells now have fatty acid phospholipids. Some, the archaea, have ethers, and I'll show it to you. They are not bacteria and eukaryotes have the membranes we think about. Archaea have very different membranes. They must have been very early after the last universal ancestor. So bacterial and elephant membranes are made of ester-linked an acid, a fatty acid. And the fatty acid is a hydrocarbon, and it ends and we have bilayers. Archaea, which are lots of cells, and they are the ancestors of our nucleus, they have ethers. Look, there's no fatty acid. And sometimes they go only a half across the membrane, so it's a bilayer, but sometimes they go all the way across the membrane, 40 carbons, about 40 carbons long, little branches sometimes in archaeolipids. And so it is a monolayer, but with two heads, a head here and a head here, okay? And so the decision to use fatty acids and esters happened after the first cell, even after the last universal ancestor. In both cases, with archaea, which have a monolayer of tetraethers instead of a bilayer of fatty acids, we now have proteins, and they are the same proteins basically in the membrane. The membrane proteins you're familiar with E. coli and the elephant, or with archaea. Okay? Maybe I'll just stop there because the bell went, and I think that's enough information. So the main points are, there was a time when the first cell happened, and it's interesting. I think to us as 21st century bioscientists.
And then at that particular stage, there's still no steroid.
Oh, steroids are very much later, much later. E. coli has no steroids, so I never even think about it. If E. coli doesn't have it, that's not needed for life, okay? No steroids needed. E. coli doesn't have steroids. Some bacteria steal the steroids from the plants or animals they grow in, the mycoplasma. But most bacteria have no steroids, and I don't need them, okay? So, given the time, it went more slowly than I thought. Lee thought I should take out the China view of the world picture. She said I shouldn't show that.
Most of the bacteria has cell wall, right?
That came later, so I didn't even talk about it. Yes,
So
and
what
plants have cell walls. Yes.
But the cell wall are different between bacteria cell wall versus plants. I'm just curious, when after bacteria come out, they just lost their originally bacterial cell wall, or they evolved independently using the ether link lipid, so they don't need.
No, well, the bacterial, their cell wall has other compounds that what's called a murin, peptidic glycan, most bacteria, not archaea. So that's later, that's not the first cell. And peptidic glycans in almost all bacteria to keep it rigid and stop it from exploding like a red blood cell would. Then gram-negative bacteria have an outer membrane, which is again a membrane, a phospholipid, bilayer membrane with proteins in it. But the outer membrane of bacteria is permeable. It has holes in it. It's not sealed like the cytoplasmic membrane of elephants and E. coli. So there is a question of whether having two membranes came very early, and then the bacteria that have only one membrane now, such as tuberculosis and bacillus, came later. But that's not the first cell. That's evolution after the last universal ancestor, the last shared ancestor. And so the origin of life people are not, do not usually ask about cell walls. It's the first time in maybe 10 times I've given this talk that anyone's asked about cell walls. It's interesting, but it's it's a different subject.
Yeah.
And of course, plants have a different kind of cell, totally unrelated, and animals don't. Okay?
The real question is whether the eukaryote lineage lost the cell will become eukaryote.
Okay, may I disagree? The real question is how did we go from no life to life? Okay. Yes, at the time that we made the eukaryote, they clearly had a cell wall, and the cell wall got lost in animals. But not in all what we used to call protozoans. Okay?
After finishing the human genome project, but then there has found an institute recently try to cook and made the first home and then make
Yeah, different
cell.
subject, but also I have never. Again, I don't want to do this kind of research. And you're talking about a synthetic cell.
Right.
Synthetic cell. Okay? And that's a totally different.
But what I wanted to mention is, interestingly, they found a couple hundred genes on essential because they remove those, and those artificial cells cannot survive or divide.
Yeah.
I think they found those genes do not include the gene for ribosome and not for mitochondria.
Okay, but
need some fatty acids.
the synthetic cell must have, the synthetic cell, and Venter, as you said, is doing it, and that is start with the cell, with everything,
Yeah.
and then make DNA on a machine, on a nucleic acid synthesizing machine. So make an artificial chromosome, and then put it inside and get rid of the old chromosome. So the only way to do this is to already have ribosomes and everything, bioenergy and amino acids inside the cell. Then you can get rid of the chromosome, and so a synthetic cell is not really is not so synthetic. I know other Taiwan universities. Will there be synthetic cells at this university? Other Taiwan universities are very interested in synthetic cells for two reasons. The government, for biofuels, they hope that we can make a very artificial synthetic cell that will make hydrocarbons so we don't need to import oil in Taiwan. And the other is to remove carbon dioxide. Europe and North America wants to remove carbon dioxide to prevent global warming. And one hypothesis as to how this might be done is by a synthetic cell. I think it is just a dream.
Yes.
Biofuels is more than a dream, but I think to remove carbon dioxide is just a dream.
At least they found some of the genes. They found probably only need a couple hundred to 1,000 genes.
Well, again, this is not the first cell which had a ribozyme, which was a nucleic acid polymerase, had one gene. The first cell had one gene, and it was called polymerase, ribozyme. Now it was already known, because Venter, before he did the human genome, his group at Tiger did mycoplasma, which had about 450 genes, and you could eliminate more than 100. So it's amazing to everyone. It only takes about 300 genes to make a cell. That's surprising. Very few genes.